15, immunological tolerance, and we are continuing with the part that is discussing about the mechanism of autoimmunity. The possibility that an individual's immune system may react against the autologous antigen and cause tissue injury was appreciated by immunologists from the time that the specificity of immune system for foreign antigen was recognized. In the early 90s, Paul Ehrlich coined the rather melodramatic phrase horror autopsies, the horror of self-toxicity, to describe the body fears of self-destruction by immune system. Autoimmunity is an important cause of disease in humans, estimated to affect at least 2 to 5% of USA population, and the incidence of many autoimmune diseases seems to be rising. The term autoimmunity is often erroneously used for any disease in which immune reactions accompany tissue injuries, even though it may be difficult or impossible to establish the role of immune response against particular self-antigen in causing these disorders. Because inflammation is a prominent component of these disorders, they are sometimes grouped under immune-mediated inflammatory disease, which does not imply that the pathological response is directly against self-antigen. The fundamental questions about autoimmunity are how self-tolerance fails and how self-reactive lymphocytes are activated. Answer of these questions are needed to understand the cause and pathogenesis of autoimmune disease, which is the major challenge in the immunology. Our understanding of autoimmunity has improved greatly during the past two decades, mainly because of development of informative animal model of these diseases, the identification of the gene that may predispose to autoimmunity, and improved method of analyzing immune response in humans. The factors that contribute to development of autoimmunity are genetic susceptibility and environmental trigger, such as infections and local tissue injury. Susceptibility genes may disrupt self-tolerance mechanism, and infection or necrosis in tissue promote the influx of autoreactive lymphocyte and activations of these cells, resulting in tissue injury. Infections and tissue injury may also alter the way in which self-antigen are displayed to the immune system and induce the production of pro-inflammatory cytokines, leading to failure of self-tolerance and activation of self-reactive lymphocytes. The role of these factors in the development of autoimmunes are discussed later. Other factors such as changes in the host microbiomes may play roles in pathogenesis and being actively studied, but this field of inquiry is still in its infancy. General features of autoimmune disorders. Autoimmune diseases may be systemic or organ-specific depending on the distribution of autoantigens that are recognized. For example, the formation of circulating immune complex composed of self-antigen and specific antibody typically produces systemic disease such as systemic lupus erythematosus. In contrast, the autoantibody and T-cell response against self-antigen with restricted tissue distribution lead to the organ-specific disease such as myasthenia gravis affecting muscle function, type 1 diabetic affecting insulin production by the pancreas, and MS affecting myelinated nerve function. So we have the diagram postulated mechanism of autoimmunity. In this proposed model of an organ-specific T-cell mediated autoimmune disease, various genetic locks may confer susceptibility to autoimmunity, in part by influencing the maintenance of self-tolerance. Environmental triggers such as infections and other inflammatory stimuli promote the influx of lymphocytes to tissue and activation of self-reactive T cells, resulting in the tissue injuries. So we have genetic susceptibility genes, then failure of self-tolerance, then after failure of self-tolerance, self-reactive lymphocytes will go to reaction to the environmental stimuli. Then we have tissue injury or inflammations also can contribute. So it's failure of tolerance, tissue injury or inflammations. Then you have tissue activated antigen presenting cells. When they meet this self-reactive lymphocyte, activation of self-reactive lymphocytes happen, and this leads to self-reactive effector lymphocyte tissue injury, autoimmune disease, because effector T cells will have different properties. Various effector mechanisms are responsible for tissue injury in different autoimmune disease. This mechanism includes immune complex, circulating autoantibodies, and autoreactive T lymphocytes are discussed in chapter 19. The clinical and pathological features of disease are usually determined by nature of dominant autoimmune response. Autoimmune disease tend to be chronic, progressive, and self-perpetuating. The reason for these features are that the self-antigen that trigger these reactions are persistent, and once an immune response starts, many amplification mechanisms are activated and perpetuated to perpetuate the response. In addition, a response initiated against one self-antigen that injures the tissue may result in the release and alteration of other tissue antigens, activation of lymphocytes specifically for these other antigen, and the exacerbations of the disease. This phenomenon is called epitope spreading, and it may explain why once an autoimmune disease has developed, it may become prolonged and self-perpetuating. Immunological abnormality leading to autoimmunity. Several immunological aberrations have been associated with the development of autoimmunity in humans and experimental models. The main such abnormality are as following: defective of self-tolerance, inadequate elimination or regulation of T and B cell, leading to an imbalance between lymphocyte activation and control, is the underlying cause of all autoimmune diseases. The potential for autoimmunity exists in all individuals because some of the clones of developing lymphocytes may express receptors specifically for self-antigen, and not all of these clones are deleted during their maturation. So, many self-reactive lymphocytes are present in healthy individuals. Also, many self-antigens are readily accessible to lymphocytes. As discussed earlier, tolerance to self-antigen is normally maintained by selection process that prevent the maturation of some of our self-antigen specific lymphocytes, and by the mechanism that inactivate or delete self-reactive lymphocytes that do mature. Loss of self-tolerance may result in self-reactive lymphocyte. Loss of self-tolerance may result if self-reactive lymphocytes are not deleted or inactivated, and if antigen-presenting cells are activated so that self-antigen are presented to immune system in an immunogenic manner. Experimental model and some results from analysis of human disease have shown that any of the following mechanisms may contribute to the failure of self-tolerance. First, defective in deletion, negative selection of T cell, B cells, or receptor editing in B cell during the maturation of these cells is in the generative lymphoid organ. Defective number of or function of regulatory T lymphocytes, defective apoptosis of mature self-reactive lymphocyte, and also inadequate function of inhibitory receptors. So any of these from down is inadequate function of inhibitory receptors, defective apoptosis of mature self-reactive lymphocytes, defective number or function of regulatory T lymphocyte, defective in deletions, negative selection of T or B cells, or receptor editing in B cell during the maturation of cells of these cells in the generative lymphoid organs. Abnormal display of self-antigen. Abnormality may include structural changes in self-antigen resulting from enzymatic modification or from cellular stress or injury. If these changes lead to the display of antigenic epitopes that are not present normally, the immune system may not be tolerant to these neoantigens, thus allowing antigen self-response to develop. Examples include citrullation of various self-peptide in rheumatoid arthritis and post-translational modification of islet proteins in type 1 diabetes. Inflammation or initial innate immune response. As we have discussed in previous chapters, the innate immune response is a strong stimulus for the activation of lymphocytes and the generation of adaptive immune responses. Infection or cell injury may elicit the local innate immune reaction with the inflammation. These may contribute to the development of autoimmune diseases, perhaps by activating antigen-presenting cells to express increased level of co-stimulators, which overcomes regulatory mechanism and result in excessive T cell activation. Cytokines made during innate immune response are also capable of activating T and B lymphocytes. An example is the production of type 1 interferon gamma associated with the systemic lupus erythematosus and other autoimmune disease. Much recent attention has focused on the role of T cells in autoimmunity for two main reasons. First, helper T cells are the key regulator for both cell-mediated and humoral immune response to protein, and most self-antigen implicated in autoimmune disease are proteins. Second, the risk of developing severe autoimmune disease is associated with inheritance of certain MHC alleles, HLA, human leukocyte antigen complex in humans, and the function of major histocompatibility complex molecule is to present peptide antigen to T cells. Failure of self-tolerance in T lymphocyte may result in autoimmune disease in which tissue damage is caused by cell-mediated immune reaction. Helper T cells abnormality may also lead to the autoantibodies production because helper T cells are necessary for the production of high-affinity antibodies against protein antigens. In the following section, we will describe the general principles of the pathogenesis of autoimmune disease with an emphasis of susceptibility genes, infection, and other factors that contribute to the development of autoimmunity. We will describe the pathogenesis and features of some illustrative autoimmune disease in Chapter 19. Genetic basis of autoimmunity. From the earliest studies of autoimmune disease in patients and experimental animals, it has been appreciated that these diseases have a strong genetic component. For example, type 1 diabetes shows a concordance of 35% to 50% in monozygotic twins and 5% to 6% in dizygotic twins. And other autoimmune diseases show similar evidence of a genetic contribution. Linkage analysis in families, genomes-wide association studies, and large-scale sequencing efforts are revealing new information about the gene that may play causal roles in the development of autoimmunity and chronic inflammation disorders. The most autoimmune diseases are complex polygenic traits in which affected individuals inherit multiple genetic polymorphisms that contribute to the disease susceptibility, and these genes act in the environmental factor to cause the disease. Some of these polymorphisms are associated with several autoimmune diseases, suggesting that the associated genes influence general mechanism in immune regulation and self-tolerance. Other locks are associated with particular diseases, suggesting that they may affect organ damage or autoreactive lymphocyte of particular specificities. Each genetic polymorphism makes a small contribution to the development of particular autoimmune disease and is also found in healthy individuals but at lower frequency than in patients with the disease. It is postulated that in individual patients, multiple such polymorphisms are co-inherited and together account for development of the disease. Understanding of the interplays of multiple genes with one another and with environmental factors is one of the continuing challenges in the field. The best characterized genes associated with autoimmune diseases and our current understanding of how they may contribute to this loss of self-tolerance are described here. Associations of MHC allele with the autoimmunity. Among the genes that are associated with autoimmunity, the strongest association are with MHC complex genes. In fact, in many autoimmune diseases such as type 1 diabetes, 20 or 30 disease-associated genes have been identified. In most of these diseases, the HLA locus alone contribute half or more of the genetic susceptibility. HLA typing of the large group of the patient with various autoimmune disease has shown that some HLE allele occur at higher frequency in these patients than in general population. From such studies, one can calculate the odds ratio for the development of the disease in individuals who inherit various HLA alleles, often referred to the relative risky. The strongest such association is between ankylosing spondylitis and inflammatory and presumably autoimmune disease of the vertebral joints and the class 1 HLA allele B27. Individuals who are HLA B27 positive are over 100% times more likely to develop ankylosing spondylitis than individuals who are B27 negative. Neither the mechanism for this disease nor the basis of this association with HLA B27 is known. The association of class 2 HLA DR and HLA DQ allele with autoimmune disease has been received great attention mainly because class 2 MHC genes are more frequently associated with autoimmune disease as well other immune-mediated disorder than a class 1 MHC genes. Class 2 MHC molecules are involved in the selection and activation of CD4 plus T cells, and CD4 T cells regulates all immune response to the protein antigen. So association of leukocyte antigen allele autoimmune disease, we have this is table 15.3. So these are things that you can read. You have disease, rheumatic arthritis, maybe AR, TID, and what allele that is responsible. These are alleles that are responsible. You can just follow them and read. So we continue. Several features of association of HLA allele with autoimmune disease are noteworthy. An HLA disease associated may be identified by serological typing of HLA locus, but the actual association may be with other alleles that are linked to the typed alleles and inherited together. For example, individuals with particular HLA DR alleles, hypothetical DR3, may show higher probability of inheriting a particular HLA DQ allele, hypothetical DQ5, than probability of inheriting these alleles separately and randomly at equilibrium in the population. Such inheritance is an example of linkage disequilibriums. A disease may be found to be DR3 associated by HLA typing, but the causal association may actually be with an co-inherited DQ5. This realization has emphasized the concept of extended HLA haplotypes, which refer to the set of linked genes, both classical HLA and adjacent non-HLA genes, that tend to be inherited together as single unit. In many autoimmune diseases, the disease-associated nucleotides, polymorphism, encode amino acid in peptide binding cleft of MHC molecules. This observation is not surprising because polymorphic individuals of MHC molecule are located within and adjacent to the cleft, and the structure of cleft is the key dominant of both function of MHC molecule, namely antigen presentation and recognition by T cells. Diseases associated with HLE sequence are found in healthy individuals. In fact, if all individuals bearing a particular disease-associated HLE allele are monitored prospectively, most will never develop the disease. Therefore, expression of particular HLA gene is not by itself a cause or predictor of autoimmune disease, but it may be one of the several factors that contribute to autoimmunity. Inheritance of certain HLA allele confer protection against developing some autoimmune disease such as type 1 diabetes and rheumatoid arthritis. The mechanism underlying the association of different HLA allele with various autoimmune diseases are not clear. In disease in which a particular MHC allele increases the risk, the disease-associated MHC molecule may present a self-peptide and activate pathogenic T cells, and this has been established in the few cases. But a quick question has not been answered in the most cases, whether disease-associated HLA molecule present the relevant self-antigen differently from HLA molecules that are not associated with the disease. When a particular allele is shown to be protective, it is hypothesized that this allele might induce negative selection of some potentially pathogenic T cells, or it might promote development of Treg, but there is no clear evidence in support of either hypothesis. Polymorphism in non-HLA genes associated with autoimmunity. The technique of genome-wide association studies has led to the putative identification of nucleotide polymorphisms, variant of several genes that are associated with autoimmune diseases, and this has been extended by more recent genome sequencing effort. Before the genes that are most clearly validated are discussed, it is important to summarize some of the general features of these genes. As stated earlier, it is likely that combination of multiple inherited genetic polymorphism interaction with environment factor induce the immunological abnormality that leads to the autoimmunity. Many of the polymorphisms associated with various autoimmune diseases are in genes that influence the development and the regulation of immune response. Although these conclusions appear predictable, it has been reinforced the usefulness of the approaches being used to identify autoimmune associated genes. Different polymorphism may protect against disease development or increase the incidence of the disease. Statistical method used for genome-wide association studies have revealed both types of association. Most disease-associated polymorphisms are located in non-coding regions of genes. This suggests that many of the polymorphisms may affect the expression of encoded proteins. Some of many genes associated with human autoimmune diseases, which have been defined by linkage analysis, genome-wide association studies, and whole genome sequence, are listed in table 15.4, and few briefly and a few are briefly described next. PTPN22, a variant of protein tyrosine phosphate PTPN22, in which adenine at position of 620 is replaced with tryptophan, is associated with rheumatoid arthritis, type 1 diabetes, and autoimmune thyroiditis and other autoimmune disease. The disease-associated variant causes complex signaling alteration in multiple immune cell population. Preciously, how these changes lead to autoimmune is not known. Node 2, polymorphism in these genes are associated with Crohn's disease, one type of inflammatory bowel diseases. Node 2 is a cytoplasmic sensor of bacteria peptoglycan. seen already in chapter 4, and is expressed in multiple cell types, including intestinal epithelial cells. It is postulated that the disease-associated polymorphism reduces the function of node 2, so it cannot provide effective defense against certain intestinal microbes. As a result, these microbes are able to transvert the epithelial and initiate a chronic inflammatory reaction in the intestinal wall, which is the hallmark of inflammatory bowel diseases seen in chapter 19. Crohn's disease may be an upregulated response to commensal microbe that has features of autoimmune disease. Complement protein. Genetic deficiency of several complement protein, including C1q, C2, and C4, are associated with the systemic lupus erythematosus. The postulated mechanism for this association is the complement activation promote the clearance of circulating immune complex and apoptotic cell bodies. And in the absence of complement proteins, these complexes accumulate in the blood and deposit in tissue, and antigen of that cell persists. There is also some evidence that complement activation increases signaling in B cells and promote tolerance, but how or even if the complement system is activated by self-antigen is unclear. Interleukin-23 receptor. Interleukin-23 receptor, some polymorphism in the receptor for interleukin-23 are associated with increasing susceptibility to inflammatory bowel diseases and skin diseases, psoriasis, where other polymorphisms protect against development of this disease. Interleukin-23 is one of the cytokines involved in the development of Th17 cells, which stimulate inflammatory reaction. CD25, IL-25R gamma polymorphism affecting the expression or function of CD25. The alpha chains of interleukin-2 receptor are associated with the MS, multiple sclerosis type 1, diabetes, and other autoimmune diseases. These changes in CD25 likely affect the generation and function of Tregs. Although there is no definitive evidence for the causal link between CD25 abnormality and Treg defect and the autoimmune disease, FC gamma RIIB, a polymorphism altering an isoleucine to threonine in the transmembrane domain of this inhibitor FC receptor, impair inhibitory signaling and is associated with the systemic lupus erythematosus in humans. Genetic depletion of this receptor in mice also result in lupus-like autoimmune disease. The likely mechanism for this is the defective antibody-mediated feedback inhibition of B cells. ATG16L1, a loss of functional polymorphism in this gene, which replaced the threonine in position 300 with alanine, is associated with Crohn's disease. ATG16L1 is one of the family of protein involved in autophagy, a cellular response to infection, nutrient depression, and other form of stress. How this polymorphism contributes to the inflammatory bowel disease is not known. Some possible mechanisms are discussed in chapter 19. Insulin. Polymorphisms in insulin genes that encode variable number of repeat sequences are associated with type 1 diabetes. This polymorphism may affect the thermic expression of insulin. It is postulated that if the protein is expressed at low level in the thymus because of genetic polymorphism, developing T cells specific for insulin may not be negatively selected. These cells survive in mature immune repertoire and are capable of attacking insulin-producing isolate B cells and causing diabetes. Although many genetic associations with autoimmune disease have been reported, A continuing challenge is to correlate the genetic polymorphism with pathogenesis of the disease. Inherited single gene, Mendelian abnormality that cause autoimmunity. Studies with mouse model and patient have identified several genes that strongly influence the maintenance of tolerance to self-antigen. Unlike complex polymorphism described previously, these single genes defect are example of Mendelian disorder in which the mutation is rare but has a high penetrance, so that the most individual carrying the mutation are affected. We mentioned many of these genes earlier in the chapter. We discussed the mechanism of self-tolerance. Although these genes are associated with rare autoimmune disease, their identification have provided valuable information about the importance of virus mechanism in maintenance of self-tolerance. The protection encoded by these genes contribute to the central tolerance, IRA generation for and function of Treg, interleukin 2 and interleukin 2 receptor, antigen and the function of Treg, CTLA4, peripheral deletions of T and B lymphocytes, antigen, and regulation of pathogenic T cells in the mucosa tissue, interleukin 10, interleukin 10 receptor. None of these mutation is seen in common autoimmune diseases. These are just selected non-human leukocyte antigen genes associated with autoimmune disease. It's good to pass through, but they are good just to have them and remember when we want to do something else. Discontinue. Then let us see role of infection in autoimmunity. Viral and bacterial infections may contribute to the development of exacerbation of autoimmunity. In patient and in animal model, the onset of autoimmune disease is often associated with or preceded by infection. In most of these cases, the infection microorganism is not present in lesions. And is not even detectable in the individual when autoimmune develop. Therefore, the lesion of autoimmunity are not due to the infection itself, but result from host immune response that may be triggered or is regulated by microbes. Infection may promote development of autoimmune by principal mechanism. Infection of particular tissue may induce local innate immune response that recruit lymphocytes, a leukocyte into the tissue and result to the activation of antigen-presenting cell. This antigen-presenting cell begins to express the costimulatory and secrete T-cell activated cytokine, resulting in the breakdown of T-cell tolerance. And specific tolerance, thus the infection results in the activation of T cells that are specific for self-antigen and not only for infectious pathogen. The importance of aberrant expression of costimulators is suggested by experimental evidence that immunization of mice with the self-antigen together with strong adjuvant which mimic microbes result in the breakdown of self-tolerance and development of autoimmune disease. In other experimental models, via antigen expressed in tissue such as isolated B cells induces T cells tolerance, but a systemic infection of the mice with the virus results in the failure of tolerance and autoimmune destruction of the insulin producing cells. Microbes may also engage in toll-like receptor on dendritic cell, leading to the production of costimulators and lymphocyte activated cytokines and autoreactive B cells, leading to autoantibody production. A role of T cell toll-like receptor signaling in autoimmunity has been demonstrated in mouse model for lupus. Infectious microbes may contain antigen that cross-react with self-antigen, so immune response to the microbes may result to the reaction against self-antigen. This phenomenon is called the molecular mimicry because antibody of the microbes cross-react with or mimic self-antigen. One example of immunological cross-reaction between microbial and self-antigen is the rheumatic fever, which developed after streptococcal infections and is caused by anti-streptococcal antibodies that cross-react with myocardial proteins. These antibodies are deposited in the heart and cause myocarditis. DNA sequencing has revealed numerous short stretches of homologies between myocardial protein and streptococcal proteins. However, significance of limited homologs between microbial and self-antigen is common autoimmune disease remain to be established. So this is normal resting tissue. Self-antigen, self-tolerance can happen. Activation of antigen-presenting cell. Expression of co-stimulators. If they're expressing a lot of co-stimulators, they cause self-reactive T cells. And this can cause autoimmunity. Or molecular mimicry. So massive activation of antigen-presenting cell or molecular mimicry can cause autoimmune disease. Role of infection in development of autoimmunity. Normally encounter of mature self-reactive T cells with the self-antigen presented co-stimulator deficiencies resting tissue antigen presenting cells, resulting in the peripheral tolerance and allergy. Other possible mechanisms for self-tolerance are not shown. B. Microbes may activate the antigen-presenting cell to express co-stimulators, and then antigen-presenting cells present self-antigens. The self-reactive T cells are activated rather than rendered tolerant. C is some microbial antigens may cross-react with self-antigen molecule. Therefore, immune response initiated by microbes may activate T cells specifically for self-antigen, which is for self-antigen. Some infections may protect against the development of autoimmunities. Epidemiological studies suggest that reducing infections increase the incidence of type 1 diabetes. Repeat. Epidemiological studies suggest that reducing infection increases the incidence of type 1 diabetes and the MS, and experimental studies show that diabetes in non-obese diabetic mice is greatly retarded if the mice are infected. It seems paradoxical that infection can be trigger of autoimmunity and also inhibit autoimmune disease. How they may reduce the incidence of autoimmune disease is not known. In the intestinal and cutaneous microbes may influence the development of autoimmune disease. As discussed in chapter 14, humans are colonized by commensal microbes that may have significant effect on the maturation and activation of the immune system. This idea is supported by finding that alteration in the microbiomes affect the incidence and severity of autoimmune disease in the experimental model. It is less clear if changes in human microbiomes affect incidence of autoimmune disease, mainly because of inherent variation and complex in microbiome individual. Other factors in autoimmunity, the development of autoimmunity is related to several factors in addition to susceptibility genes and infection. Anatomic alteration in tissue caused by inflammation, possibly secondary to infection, ischemic injuries, and trauma may lead to the exposure of self-antigen that are normally concealed from the immune system. Such a sequential antigen may may not have reduced the self-tolerance. Therefore, previously the hidden self-antigen are released. They can interact with immunocompetent lymphocyte and induce specific immune response. Example of the anatomical sequence study amino antigen is the so-called immune privileged tissue, including intraocular and intracellular proteins. Post-traumatic uveitis and otitis, which can be bilateral even when trauma is unilateral, are thought due to the autoimmune response against self-antigen in that is released from the normal location by trauma. So gender plays a role as well. So a role in several autoimmune diseases. Many autoimmune diseases have high incidence in women than men. For example, SLE affects women about 10 times more frequently than men. The lupus disease, mice develop only in female, and is secreted is female and is predominant result from the influence of sex hormones or other gene-related factors. Although autoimmune disease are among the most difficult scientific and clinical problems in immunology, the current knowledge of pathogenic mechanism remain incomplete. So theories and hypotheses continue to outnumber fact. The application of new technology advances in rapid improving understanding of self-tolerance will and is hoped to lead to clear and more definitive answer to enigmas of autoimmunity. Summary. Immunological tolerance is unresponsiveness to antigen induced by exposure or specific lymphocyte to that antigen. Tolerance to self-antigen is a fundamental property of normal immune system. And failure of self-tolerance leads to autoimmune disease. Antigen may be administered in a way that induces tolerance rather than immunity. And this may be explored for prevention and treatment of transplant rejection and autoimmune and allergic disease. Central tolerance is induced in the generative lymphoid organ, thymus, and the bone marrow. When immature lymphocytes encounter self-antigen present in these organs, peripheral tolerance occur when mature lymphocytes recognize self-antigen in peripheral tissue. And in T lymphocyte, central tolerance occur when immature thermocytes with high affinity receptor for self-antigen recognize this self-antigen in thymus. Some immature T cells that encounter self-antigen in the thymus dies. Negative selection and other develop to FOX. FOX regulatory T lymphocyte Treg that function to control response to self-antigen in peripheral tissue. Several mechanisms account for peripheral tolerance in mature T cells. In CD4 T cells, energy is induced by antigen recognition without adequate co-stimulation by engagement of inhibitory receptors such as CTL4, cytotoxic T lymphocyte antigen 4, and PD1, the programmed cell death protein 1. Treg inhibit immune response by multiple mechanisms. T cells that encounter self-antigen without other stimulus or that are repeatedly stimulated may die by apoptosis. B lymphocyte central tolerance is induced by immature B cell recognize multivalent self-antigen in bone marrow. The result is the acquisition of new specificity called receptor editing and apoptotic death of immature B cell. Mature B cells that recognize self-antigen in the peripheral in the absence of T cells may be rendered anergic and ultimately die by apoptosis and become functional and responsive because the engagement of inhibitory receptor. Autoimmune results from inadequate self-tolerance regulation of lymphocyte. Autoimmune reaction may be triggered by environmental stimuli such as infection in genetical susceptible individual. Most autoimmune diseases are polygenic and numerous susceptibility genes contribute to this development. And the greatest contribution is from major histocompatibility complex. Other genes are thought to influence the selection or regulation of self-reactive lymphocytes. Infection may predispose to autoimmune by several mechanisms, including enhanced expression of co-stimulation in tissue and cross-reaction between microbial antigen and self-antigen. Some infection may protect individual from autoimmune by unknown mechanism. Yeah. So that's the end of this chapter. There are a lot of reading that you can go through. So the next chapter is chapter 17 because chapter 16 was already covered.